And welcome to the fourth and final presentation of the 2021 Fish Tales Lecture Series, presenting the science of Great Lakes fisheries. Tonight's presentation is the Great Lakes Mass Marking Program for Trout and Salmon, Better Fisheries Through Better Science by Charles Bronte. My name is Mark Holey, and again, I will moderate tonight's lecture. The Fish Tales Lecture Series is a collaboration between myself, a retired fish biologist, and Kagan Haringa, the naturalist and program director for the Crossroads at Big Creek Learning Center and Nature Preserve. And we welcome your comments about the lecture series and any suggestions you may have for future topics. So to provide your feedback, please send an email to fishtaleslecture@gmail.com as shown on the screen. I also need to recognize our partnership with the Door County Library and Laura Kayakan as our contact and for their expertise and support to host the 2021 Fish Tales lectures via Zoom and Facebook live presentations. You can actually access copies of any of the 2021 lectures by visiting the Door County Library calendar online, find the date of the lecture you're looking for, and click on the video link. Or if you send an email to fishtaleslecture@gmail.com, I will share the YouTube link with you. For the courtesy of the speaker and the audience, please make sure that your microphone remains muted and turned off during the presentation. We look forward to your questions, so please enter them by using the chat feature in the bottom toolbar on your screen. We will review all the questions asked at the end of the presentation. You know, the invasion of the sea lamprey in the 1940s ecologically changed the fisheries of the Great Lakes forever. Before the basin-wide sea lamprey control program that we heard about at our previous Fish Tales lecture was initiated, lake trout, the native top predator of the Great Lakes, was extirpated from Lake Michigan, which in part allowed the non-native alewife to invade and rapidly increase in number in Lake Michigan. Unchecked by predators, the alewife explosion culminated in 1967 by the nationally publicized die-off that was estimated at hundreds of millions of pounds. Stocking Pacific Chinook and coho salmon was one of the actions management agencies implemented to utilize the surplus alewife as bait to create a fishery, a sport fishery, and what a world-class fishery it became. During the first 20 years of the Pacific salmon fishery, the more salmon the state fishery agencies stocked, the more salmon anglers caught. That salmon fishing bonanza ended suddenly in the late 1980s and early 1990s when the Chinook salmon abundance declined by an estimated 50% and the average size of salmon declined drastically as well. To adjust to the decline of Chinook salmon and balance the restoration and management of all trout and salmon in Lake Michigan, fishery agencies recognized the need to develop new tools to provide the level of data required to address the new fisheries paradigm in Lake Michigan. Probably the most important tool that has been developed and implemented has been the Great Lakes Mass Marking Program. Tonight, we are pleased to have Chuck Bronte, the director of the Great Lakes Fish Tagging and Recovery Lab of the U.S. Fish and Wildlife Service, to present how the mass marking tool has informed the management of trout and salmon in Lake Michigan and the Great Lakes. Chuck is a senior fishery biologist for the U.S. Fish and Wildlife Service at the Green Bay Fish and Wildlife Conservation Office, where he directs research and restoration planning for native fisheries in the Great Lakes. He was the principal architect of the Fish and Wildlife Service's large-scale coded wire tagging and data recovery program that assists the states and tribes with managing salmon and trout sport, commercial, and treaty fisheries and evaluates progress toward lake trout restoration. Chuck has published over 90 papers on fishes of the Great Lakes, primarily dealing with biology and evaluation of the recovering lake trout populations. He has served for the past 20 years on several research boards of the Great Lakes Fishery Commission and on editorial boards for the Transactions of the American Fisheries Society, the North American Journal of Fisheries Management, and the Journal for the Great Lakes Research. He holds a master's degree in biology from Murray State University. Chuck, we're looking forward to your presentation. So take it away and tag, you're it.
Okay, thanks, Mark. And thanks, Kagan, and everybody else that's invited me and putting on this series. I appreciate the introduction, Mark. You've probably taken a third of my presentation already, but I'll run through that introductory material very quickly here. And just so folks know, Mark was my boss for what, 20 years or so at the Green Bay office. So what Mark was alluding to was some of this historical information that is important as the backdrop, and I'll go through it very quickly. Historically, let's say before 1950, we had lake trout, the principal predator, which was true in all the Great Lakes, and supported by a prey base of white fishes or corrigonian fishes that were like no other in the world, up to 10 different forms. Basically, the lake trout ate these white fishes, and that supported the largest lake trout populations and fisheries in the world. This also supported earlier on Aboriginal subsistence fisheries, and then later large-scale commercial fisheries in the 1800s and through the last century, until overfishing combined with exotic species like sea lamprey, alewives, and rainbow smelt, which are not native to the Great Lakes, disrupted these communities. At that point, fishery management was very limited, basically year restrictions and season restrictions. And there was stocking done, which began back in the late 1880s, but that really did not, was very ineffective. Current state, or more recent times, is we have lake trout had collapsed, and now we stock them for restoration purposes in most of the Great Lakes, and that's supplemented by salmon and brown trout, which were stocked to not only to control the non-native alewives and utilize that resource to create and diversify fisheries. There was little natural reproduction by lake trout outside Lake Superior, and there was now unknown levels of natural reproduction by most of the salmon, the non-native species that were stocked by the states. We have much limited commercial fisheries now, mostly concentrated on lake whitefish. We have newly developed treaty fisheries, which are executed in 3 of the upper Great Lakes, and now a very economically important and culturally important salmon fishery centered mostly around Chinook salmon. Contrary to pre-collapse times, fisheries management is now relatively intense. We spend millions of dollars controlling sea lamprey and millions of dollars stocking and salmon and other species to create a $7 billion sport fishery. So basically what we have now is a very perturbed situation where most of the unique native whitefishes in Cisco, which are pictured in the upper right-hand corner here, are mostly gone. And this is what supported most of the predators historically. Most of the lake trout, of which there were numerous forms that had different ecological roles and lived in different places and looked very differently, are virtually gone throughout the entire Great Lakes, with the exception of Lake Superior. Non-native alewives and rainbow smelt, in the absence of lake trout, expanded and were left unchecked by the lack of predation, which caused those massive die-offs that Mark referred to, which most of us remember when we were much younger. Sport fisheries were diminished, and some of the largest commercial fisheries in the world had collapsed and certainly did not bode well for the economies of the shoreline communities. So everyone started stocking salmon. In addition to lake trout, which was the role of the federal government, the U.S. Fish and Wildlife Service was stocking those fish not only to provide fisheries, but for restoration purposes. The states were involved in stocking brown trout, steel, coho salmon, and Chinook salmon, again, to diversify the sport fisheries and take advantage of the large standing stock of alewives and rainbow smelt that was available. The bioenergetics of these non-native species could actually rapidly use the standing stocks of alewives that were there. Lake trout have a much longer life history. Their bioenergetics, that is how much they consume in relation to how they grow, is much more slower and really could not control alewives the way these other non-native species did. So now this led, as Mark led up to in his introduction, the need for the coded wire tag program. And this is sometimes referred to as mass marking meaning all the fish are marked or tagged in mass before stocking out. And I define it as a comprehensive, coordinated fish tagging and marking, and more importantly, a data recovery program involving all agencies that stock salmon into the Great Lakes and trout. And what does it do? It provides tagging and marking services for the over 12 million fish that are stocked by state and federal hatcheries across the Great Lakes Basin, and more importantly, a system to collect, process, and cooperatively analyze the return data to assist the agencies in evaluating the impact of their programs, and for the federal government to look at the rate of recovery of native lake trout, which is a goal of the federal government. This idea was first suggested by the Council of Lake Committees of the Great Lakes Fishery Commission, which directed a multi-agency task group to investigate mass marking technology for a program basin-wide. And I was asked to co-chair, be the US co-chair of that group. And this occurred, well, more than 10 years ago. Most of the states and tribes, you can see in the lower left here, most of the states and tribes that have management authority are represented on the Council of Lake Committees. So why does the Great Lakes need this program? Well, previously, most fish that were stocked into the Great Lakes, with the exception of lake trout, were stocked with no tag or mark. We had no idea who was catching whose fish, what the fate of these fish were, what the performance were. There were previous tagging programs used for mass marking, basically using coded wire tags, but these were limited in scope and usually carried out unilaterally by one agency or a few agencies on Lakes Michigan and Huron, and that really doesn't get you the right answers to the important questions. Management requires much better information on the performance of hatchery-reared salmon and trout, and that's something that mass marking can provide. And certainly the fish is too valuable. As I said, it's worth $7 billion on the US side alone. And we need better information to make management decisions. So what information is gained by this program that wasn't available before? Well, first of all, levels of natural reproduction by non-native salmons and also lake trout. Basically, those fish that don't have a tag or a mark are construed to be wild lake-produced fish, especially if you're marking and tagging all the fish that get stocked into the lake. We need to know about interjurisdictional movement of stocked fish, who's catching whose fish and where and when, and what their contributions to the different fisheries are. We want to be able to identify fish hatcheries and strains of fish and sizes of fish that have the greatest returns to the fishery, which is something that's a very important biological, but also a business measurement. And we get more accurate population metrics on the fish that we recover, like growth rates, survival, exploitation, and year class membership, which gives us how old the fish are. So why is it important? We got a $7 billion fishery. And then on top of that, we got a $12 billion boating industry. These types of economic activities deserve the investment that they get now. We had stocking that was ongoing with that's now putting increased pressure on forage fishes, which are in decline. And this makes the fishery management outcomes unpredictable. So it's best that we're able to estimate how many predators we have in the lake, not only the ones that we're stocking, but the one that's lake producing. And that's what the mass marking program can do and is doing now. And we need to know the wild fish of native species of restoration and conservation significance. We must distinguish them from their hatchery counterparts so we can actually measure the progress in restoration. And interjurisdictional populations, which these fish are, require basin-wide coordinated effort. And it's the only way to get the right answers to manage this important fishery. So what is a coated wire tag? Perhaps those anglers that are listening in are familiar with this program. Coated wire tag is a 1 millimeter piece of metal that has a 6-digit code that's laser etched into that piece of metal, and that metal is inserted into the nose of the fish while they're in the hatchery. And along with that, they get an adipose fin clip, which is the universal sign that the fish may have a coated wire tag in their nose. The code is unique to a specific group of fish that share the same attributes, like the agency that stocked it, the hatchery it came from, its stocking location, its genetic strain, size at stocking, life stage, et cetera, et cetera. So by looking at the recovery of all those fish over time that share the same code and analyzing that together, we can make some determinations on the performance of fish and the attributes of the fishery. So how do we do this? Well, we tag millions of fish a year, and we have to do that rapidly and efficiently. And so we have these automated fish tagging trailers that were developed by a company in the Pacific Northwest, which we actually modeled this program based on what was going out in the Pacific Northwest for salmon management that's been going on for over 35 years. These are automated tagging trailers that are computer operated, that automatically sort the fish by size. They measure each fish to 1/10 of a millimeter, sort them, and then clip the adipose fin, which is the fleshy fin at the just forward of the tail, and then inserts a coated wire tag into its nose. We have 4 of these trailers. Each of them are worth about $1.4 million and probably more today to replace them. And these go from, these trailers travel from state hatcheries to federal hatcheries all around Lakes Michigan and Huron right now, which is the center of operation for this program at this point. And they tag up to 10 million fish a year. We can process these fish on the average, up to 8,500 fish an hour. This is the interior of the trailer that you see here. and you see 6 different tagging lines here. The fish are sorted in this upper chamber here with cameras that measure the fish, and then they get diverted down to eat one of these 6 tagging lines, which is set up for different sizes of fish. The fish are never anesthetized, they're never dewatered, and they just move through this at a fairly rapid rate. We get great tag retention. 99% of the fish are clipped. That's far superior to manual methods than we used to get. And now we get accurate counts of the fish, which prior to mass marking were fairly difficult to do with the old methods that were done in the hatcheries. So here's the history of the program. We started tagging fish in 2010. And just so you know, the species that we tag are selected and approved by the Council Lake Committees of the Great Lakes Fishery Commission. It's basically the management agencies, which does not include the Fish and Wildlife Service. We do not have management authority on the Great Lakes. We provide management assistance to states and tribes. But the states and tribes decide which species get marked, and this is the history of that marking program. You can see we're doing about 9 million fish a year on average. Unfortunately, last year with COVID, we didn't do any tagging or marking, but you can see we've run the fish through run a variety of species through the tagging units, with the understanding that the initial application was concentrated on Chinook and coho and Steelhead in the Pacific Northwest, and we had to do some modifications to the unit to get it to run the additional species, especially lake trout. So the tagging program is one thing. The most important part of this is the recovery program, the data and tag recovery operations. So what we do is we actually staff up to 10 to 13 technicians a year are all around Lakes Michigan and Huron. And they're located at state partner offices that assist us in directing their activities to tournaments and fish cleaning stations and places where we can interface with anglers over the fishing season to measure their catch and process their catch to recover not only the tags in the hatchery fish, but also collect data on their wild counterparts. One of the major objectives of this study, of this program, was to estimate wild recruitment of Chinook, lake trout, steelhead, and all the other species that are being focused on by the Council Lake Committees. Nobody fields this many people to collect this much data around Lake Michigan and Huron. Today, we've processed well over 100,000 tags, and this provides us a wealth of information. The tags, as I showed you, were lodged in the rostrum of the fish. When the fish are captured by the anglers, our technicians cut off the snout. Those snouts get frozen, they get sent back to our lab, and the tags are extracted and read and put into a large relational database that holds the recovery information, the stocking information, all the biological attributes of that fish, and allows us to do the analyses that I'm going to show you in the next couple of slides. So the data analysis and support for fishery managers, as I spell managers wrong. So the research objectives, as I said, are largely determined by state and tribal partners who have the management authority. These results are regularly shared with managers and stakeholders through reports, presentations, and other outreach. We go around Lakes Michigan and Huron, speaking to a lot of sportsman groups, showing them some of the information that I'm going to share with you today. So, coated wire tags, actually the recovery of coated wire tags, has helped answer questions about wild recruitment. When I say recruitment, wild reproduction, where fish move in relation to where they're stocked, their relative survival, which is a measure of performance after they're released, and unambiguous information on growth. The data has also contributed to many mathematical models that are used to evaluate harvest levels and inform stocking decisions. These models help generate population absolute stock sizes and other metrics, which are used to basically inform how many wild fish are out in the lake, and therefore how much those fish consume of prey, therefore leading to stocking decisions that do not allow us to overtax the forage base. So the field survey environment that we have has also been used to leverage on a lot of collaborative studies that would not be possible since the sources for the data and the specimens can't be secured otherwise. So right now I'm gonna show you information on lake trout, Chinook salmon, and some of the results from the leverage studies. I'll handle lake trout first. As you know, Mark introduced the idea of the concept of the collapse of the lake trout populations. These were the largest lake trout populations in fisheries in the world. Sea lamprey and overfishing contributed most to this decline. We began stocking in Lake Michigan and stocking in all the other lakes at different timeframes with a variety of different genetic strains that were available either from Lake Superior or from Lake Michigan remnants that were moved out to Montana and Wyoming. This Lewis Lake strain was originally from Lake Michigan prior to the collapse. That was moved out to Wyoming and the Yellowstone region and stocked into the lake there. And we have gone back and grabbed that strain and have been reintroducing it into Lake Michigan. Excuse me. The Green Lake strain is also a remnant Lake Michigan genetic strain that was stocked into Green Lake, Wisconsin, and we use that as well in the early years. Several strains from Lake Superior, which did not lose all its lake trout like Lake Michigan, are also used. This Perry Sound strain is from Lake Huron, which survived sea lamprey and overfishing. And this Klondike strain is a deep water humper type lake trout from Lake Superior that was introduced in the last several years to add additional genetic diversity and the ability for this strain to occupy deep water. So you can see we've done a fairly good job of diversifying the genetic strains, and now we're in the position to coated wire tag these individual strains and follow them through time to determine which ones not only survive the best in the lake, but also later on, which produce the most wild fish when they start reproducing. So lake trout management objectives and I'm going to concentrate mostly on Lake Michigan here. Basically monitor the progress towards self-sustaining lake trout populations. The questions are lake trout recruiting naturally? Are they reproducing naturally? And where are they doing that? Recruitment by lake trout has been limited of these stocked fish has been limited by overall low population size in the past. We had spawning stocks that were too young and too few and too few in abundance. And we have early mortality of eggs and young from negative interactions with alewives, and I won't get into that very much. The way we can tell a stocked and a wild fish is by looking at whether it has a fin clip or a coated wire tag. That would denote it'd be a hatchery fish. Prior to the coated wire tag program, we would rotationally clip each year class with a different fin clip of lake trout. Now we coated wire tag since the 2010 all lake trout that are stocked across all the Great Lakes. Those fish that don't have a clip or a tag are construed to be wild. So here is a graph of the percentage of the lake trout that we observe in the sport fishery from our program that were of wild origin, those that did not have a fin clip or a coated wire tag. As you can see, things in Lake Huron are doing fairly well. Most of the population there is now composed of wild fish, although at much lower levels than they were historically, but this is a great result. Lesser success has been seen in Lake Michigan, but where we were about 15, 20 years ago when I started on Lake Michigan, this is far better than anything we had back then. Most of the wild fish are found in southern Lake Michigan. As you can see, we have almost 40 to 50% in some locales, far less in the north. So if we can look at this progress over time, this is through our sampling. We have a time series of the percent wild fish in Lakes Huron, southern Lake Michigan, and northern Lake Michigan. We can see these trend lines have been going up, mostly in southern Lake Michigan and in Lake Huron. Very, very good results. Basically, Lake Huron now has quite a few wild fish that they have been pulling back on stocking, and we hope to achieve the same thing in Lake Michigan. Northern Lake Michigan still has a problem because right now populations there are very young. They do not survive well because of sea lamprey coming out of the Manistee River and a very active fishery up there. In terms of relative abundance, we see the same trends. This is a better indication of wild recruitment or success. We can see Lake Huron is going up, and this is basically relative abundance measured in the sport fishery. This is the catch of wild lake trout in the sport fishery. Again, far fewer positive results in northern Lake Michigan because of the high mortality there. Another objective is to evaluate the efficacy of the lake trout stocking program. Questions there center around how well do lake trout survive at different stocking locations, and what genetic strains actually survive better. What strains contribute the most to natural recruitment? And we can tell this by looking at the genetic contribution of the different strains in the wild fish that we capture. So this was a study we did, we just published it last year, where we're looking at the relative survival of four different locations. This is all strains combined. We can see that Julian's Reef, which is located down in Illinois waters, has one of the highest, best survival rates combined with the Southern Refuge, which is a no fishing zone in the southern part, which encompasses some historically important spawning reefs. Lesser success is seen in the Northern Refuge, as I mentioned, where you have egregious sea lamprey mortality and a higher fishing mortality. So this basically shows us the performance of these fish in different zones, and we can relate that to the different fishing conditions there. Survival also varied among strains. So the left-hand figure shows stocked fish at Julian's Reef, which is in Illinois waters. We can see the Lewis Lake strain, which is the remnant Lake Michigan strain, does very well on these year classes that we've been able to observe in the sport fishery, compared to the Seneca Lake strain, which is a strain that was introduced from the Finger Lakes, which was perceived to have better survival advantage against sea lamprey predation, because they are able to avoid sea lamprey by living in a different area of the water column. When we go to the northern refuge, we see something very different, where we have high sea lamprey mortality. The Seneca Lake strain has done generally better than the remaining strains. And it just goes to show you that these remnant Lake Michigan strains do as well or better, especially when sea lamprey predation is at the lower levels. We did a cooperative research with Wes Larson from UW-Stevens Point, where he looked at the genetic origin of the wild fish we were recovering. And basically he showed that the Seneca Lake strain was overrepresented in some of the areas, most of the areas in the lake where they were stocked, or even where they weren't stocked. And then did a much better job than most of the other strains. Lewis Lake strains were on par with what was expected, while the Green Lake and Superior strains basically underperformed in contributing to natural reproduction. So we basically have been discontinued the Green Lake strain, kept the Lewis Lake and Seneca's Lake strain, and a few others based on the results. This is something that the Lake Committee is now reviewing for the lake trout restoration program. So another objective of the lake trout management program is to define stock assessment units, look at movements between the units, and determine which fish contribute the most to fisheries. The specific questions are, how much do lake trout move after they're stocked? And do different strains exhibit different movements? So this graph shows basically the cumulative proportion of the lake trout caught from the distance from stocking location. And this basically, this is for 3 different areas of Lake Michigan. As you can see, right around 75 to 80%, or within 62 miles or so, basically you're catching most of the fish. These fish do not move like Chinook salmon, which I'll show you a little later. They stay pretty much within about 60 to 70 miles of where they're stocked, with most of the fish staying even closer. This is consistent with virtually every other mark recapture study done for lake trout, not only in Lake Michigan, but in Lake Superior and elsewhere. So, this figure here shows fish that were stocked in the shaded area, which is MM5, which is a management unit in Michigan waters. And the percentages reflect the percentage of fish that were stocked in that unit, but that were caught elsewhere. You can see most of the fish, 48%, were stocked in the area where were caught in the area where they were stocked, with more fish in adjacent areas. But you can see most of the fish stay locally. If we looked at Indiana waters, 55% of the fish recovered were stocked there, with lesser percentages stocked elsewhere. So lake trout basically do not move extensively. And I'll show you some information on Chinook salmon that will show something very different. I'm going to move on to here. So, right now the lake trout restoration program stocks a majority of the fish on the offshore areas, the northern refuge and the southern refuge. These were historically important spawning areas prior to the collapse. This is where most of the fish or the natural reproduction occurred. There was natural reproduction in nearshore areas alone, but most of it was offshore and contributed to fisheries throughout the lake. And they still do. The fish that we stock in the offshore refuges, which are expressed in these different shades of blue, we're looking at the percentage of angler catch that's contributed by the fish that are stocked at Southern Refuge, Julian's Reef, and the Northern Refuge, all offshore locations, and in the red, which are nearshore locations. So even though we stock most of the fish offshore, they are by far contributing most of the lake trout caught by anglers. And this is likely due to higher survival in these offshore locations, which is basically shown in this bar graph here. This basically, we're seeing these blue areas. The higher the bar, the better the relative survival compared to the nearshore stocked fish. So there is a reason why we send them offshore. We want them to survive better. When they survive better, they get older, they reproduce more, and the probability of wild recruitment increases. But it's not at the expense of providing fish to the nearshore fishery. So this is showing the differences between two strains of lake trout, just to show you the differential behavior of some of these fish. We argued for the Klondike strain to be introduced from Lake Superior into Lake Michigan. In fact, it was Mark Holy who fought for this very hard early on. And the idea there is that we would get a deep water form of lake trout that would basically stay in deep water and occupy that part of the lake. And the Klondike strain actually has done that. Of most of those fish were stocked in the Southern Refuge, and only about 3% were actually found outside of the Southern Refuge. So they stayed put where we thought they would be, and that's the kind of behavior we see that they have in their native Lake Superior. On the other hand, Seneca Lake strain, which were stocked in the Southern Refuge, 72% were found elsewhere. They were not in the Southern Refuge. So very different behavior depending on the genetic strain. And we have analyses for the other strains as well. But I just wanted to promote or show you these differences. Moving on to Chinook salmon, which is what most people are interested in these days. Some of this information we've gone through. These were basically stocked to diversify the sport fishery, which was almost non-existent. It created almost an overnight fishery. Dr. Howard Tanner from Michigan led the charge on that. Other states followed around Lakes Michigan and across the basin. This was to control the dead alewives, which had no predators. These non-native forage have now declined in abundance, more likely due to the combined effects of predation, but also the lack of the lowering productivity that we're seeing in the Great Lakes with the onset of quagga and zebra mussels, and of course less phosphorus going into the system. So having these low levels of alewife and rainbow smelt, the last thing we need to do is stock too many salmon on the wild fish that, until the mass marking program was developed, we had no idea how many Chinook salmon were being lake produced. So the objective is to balance salmon abundance and prey abundances to avoid collapsing the forage base. So we need to know how many Chinook salmon enter the fishery each year. We know how many hatchery fish go in, but how many enter the fishery from wild recruitment? How stable are their growth rates? Salmon grow very quickly. Their growth rates are a great indicator of what's going on in the system and what that predator-prey ratio looks like or should look like. So again, we can tell a wild fish from a hatchery fish by looking for the adipose fin clip. You anglers can do the same when you're catching your fish, and also look for the evidence of a coated wire tag, which only we can do. So knowing these ratios of wild to hatchery fish by year class, we can actually estimate the proportion of each year class that was produced that were wild. So we've done this with coated wire tags since 2010. Prior to that, we used tetracycline, which was applied to the feed to salmon in the hatchery to create a mark in their ear bone or in their vertebrae. And anyway, we have these estimates of the percent wild across the year classes. The year classes is the year that the animal was born. As you can see, it's ranged anywhere from less than 40% all the way as high as 65%. And then we can use those ratios of wild to hatchery to estimate the entire number of smolts, both hatchery, which are in orange. We know that number for a fact, and we can estimate the wild smolts so we understand the size of the year classes that are produced every year. This is valuable management information that is now plugged into population models that help estimate total standing stock of Chinook salmon through time, but also, better yet, are combined with other models that estimate the consumption of the salmon at large that help the states manage the predator prey ratio to keep that within a range of observations that does not overtax the alewife prey base. So we measure the number or the percentage of wild fish that we see in the sport fishery. And basically right now in Lake Michigan, 68% of the Chinook salmon that you catch are wild. They don't come from a hatchery. They come from the lake. Same thing is going on in Lake Huron as well. They've been at, wild fish have been contributing to their populations for far longer. And you can see these percentages vary depending on where you're going in the lake, but they're pretty similar everywhere. So hatcheries, to some extent, are no longer really contributing to the total population. It's the lake that's kind of taking over. So what's interesting about these salmon is we can calculate or estimate their growth history. And no matter how we look at the data, this figure shows the total length in inches by the ages in years. Salmon only live to be about 5 years old. Most of them only live to 3 or 4 years old. But no matter where they're stocked or where they're recovered, they all grow the same. So you're wondering, how can that be? Well, that's because they're all moving around in Lake Michigan and Lake Huron. It's one big fishbowl. And basically, if you're staying in one location and just preying on the forage base there, you would see differential growth. But since they're moving all around the lake continuously throughout their lives, they're all growing at the same rate. And that growth varies over time and is linked directly with the alewife density. So this is the average length of an age 1 Chinook salmon at the end of its growing season with the alewife density for that year, and you see they correlate fairly well. Chinook salmon principally eat alewife. They're obligate alewife eaters. So whatever's going on with alewife, you'll see expressed in lake trout growth. Another management objective is to look at, evaluate Chinook salmon stocking rates. Does stocking location affect salmon survival? That's a great question. And here we've estimated it for, on the x-axis is a variety of locations where salmon are stocked by jurisdiction. The height of the bars indicate relative survival. High relative survival is a higher bar. And you can see that for Wisconsin anglers, we're in the sweet spot here. Wisconsin fish survive much better on average, depending on location, compared to Michigan, Indiana, and Illinois fish stocking events, much higher, and even higher than Lake Huron. So it's this kind of information that the states could use to eliminate stocking locations where they're not seeing the good returns on those stocking events. So we've looked at this across a variety of years, and this map basically shows areas that have consistently high survival regardless of year, and that's in WM 5 and 4, sometimes high WM 6 in the light blue, a kind of a boom or bust in purple, and consistently low stocking, or rather survival in the red zones. You can see Green Bay, Northern Lake Michigan, and MM6 in Michigan waters are not, have consistently low survival for the fish that were stocked there. So basically those potential factors, while we haven't tested for them, we're suggesting are related to temperature, food availability, at least in the early years, evidence of wild recruitment, and maybe predation by standing stocks of predators there. So we also want to determine what fish contribute most to fisheries. So how much do Chinook salmon move after release? I mean, that's a great question. Where are the wild fish coming from that we're seeing in the fishery? So this map, I'll pay attention to the one on the left-hand side where we're looking at, you'll see a star, an orange star by Manitowoc there. This portrays the origin of the Chinook salmon that were landed at Manitowoc. As you can see, the red dots are proportional to the proportion of fish that made up the catch at Manitowoc. As you can see, the Chinook salmon stocked at Manitowoc or in that management zone contributed very little to the open water harvest during the summer, basically June, July, August, and early September. Fish were represented from all the areas of Lake Michigan and even Lake Huron. Another example is at Frankfurt, Michigan, the figure on the right. Again, very few fish. Of course, they don't stock any fish in front of Frankfurt, but very few fish were from adjacent zones there. Fish from all over Lake Michigan that were stocked all over in Lake Michigan contributed to the fishery, and even fish from Northern Lake Huron. We have this data for over 30 different ports, and it pretty much shows the same thing. This suggests that regardless of where the fish were stocked, those fish could be captured at virtually any location. So the probability of you fishing at your favorite port and catching a fish during the open water season, a Chinook salmon that was stocked there, is pretty damn low. So you probably ask, well, we know salmon come back to where they were were stocked. And this figure shows that. We're showing the percent of fish captured that were captured within the month across time. And we can see that the fish that were stocked nearby, by the time spawning occurs in late September, most of the fish that were stocked there are spawning there in the spawning area. And this is what salmon do. And if they were stocked, and we can see that the wild fish are actually returning, moving this, and these are recoveries in Wisconsin. They're actually, the incidence of wild fish is lessening over time as the spawning season progresses. And this was data from Wisconsin only, and basically shows you the transition of wild fish moving away from Wisconsin into other waters, where we think it's in Michigan waters, because that's where they have the best spawning habitat. But even though the probability of catching a fish stocked in your area is low during the open water season, these fish are returning to an area close to where they were stocked, and likely the stream where they were stocked. So we want to look at where the wild fish are coming from. We're looking at the proportion of fish that were captured in sport fishery across months in Michigan and Wisconsin. And we can see, again, that the wild fish in Wisconsin decline over time as you get into the spawning season, where in Michigan, it's the inverse, which again suggests that most of the wild fish are coming from Michigan. And basically, they have the spawning habitat and the more appropriate stream conditions than we do along the Wisconsin shoreline. So our collection network, does help us do a lot of collaborations with other universities and states and federal government and tribes. And these are studies that rely on our collection network for specimens and data that can't be collected by other means. So this gives you an idea of the range of where we collect data across. We visit all these ports a year, while fishery independent surveys do a good job on lake trout and prey base. Our sport fishery survey provides an unparalleled ability to collect data and specimens on a variety of species across time and space. So one of the things we've been working on with a variety of folks is looking at the study of the diets of the principal predators in Lake Michigan, and we're actually doing it in Lake Huron as well. As you can see, here's the proportion of the total diets by brown trout, Chinook, coho, lake trout, and steelhead. As you can see, the blue is alewife. Everybody eats alewife. But what's important here is to look at how obligated Chinook and coho salmon are, but also look at the diversity of steelhead and lake trout, and even brown trout. Basically, those three species are able to shift their diets somewhat, a little bit more fluid than, let's say, Chinook salmon and coho salmon. But even though alewives are at low numbers, they're still contributing to the diets of virtually all the salmon and trout predators. And we can also look at this from a long-term perspective. While this looks kind of confusing, this is a biplot of the stable isotope space for nitrogen and carbon for the five principal predators. And basically, this is kind of an index of where they're feeding in their kind of trophic niche in Lake Superior. As you can see, lake trout stands out from pretty much all the rest of the predators, but also brown trout. And that reflects that diverse diet, more diverse diet that they have, while Chinook and coho and steelhead seem to be overlapping quite a bit more. But this gives kind of us an indicator on that unique niche space that lake trout have, and it's their ability to move from different prey bases. And that's what this animal does. If you look across its range, they can live in lakes with no fish and just feed on plankton, or they can live in lakes with strictly one forage fish. They can eat each other. They can do everything. And it's an amazing animal, whereas the Pacific salmon are less opportunistic. This is, we also do work on steelhead. We're actually tagging and recovering steelhead now. I don't have much data there, but we're working collaboratively with Michigan DNR and Central Michigan University, looking at where the wild steelhead come from. And we can tell this by looking at the trace element profiles in their ear bones. We know what that trace element looks like in the young steelhead that come from the streams in Michigan. And then we recapture those fish when they're older, then when they're caught in the fishery, and then we can basically match that up with the reference sample and determine where they're being produced in the Michigan streams, which is certainly important from a management perspective. The size of the circles there indicate the proportion of fish that were in the sport fishery that came from those streams. So we're doing the same thing for Chinook salmon, and now there's a coho salmon proposal as well being submitted to funding agencies. So we also completed a study where we're looking at the sea lamprey marking rates, and this gives us an idea of what the level of predation may be on these different predators. Lake trout was thought to be the principal prey of sea lamprey, and we can see on the left-hand side that in Lake Michigan, that's still the case. We see much higher rates of wounding and these are classification distributions that we classify the wounds as. Type A wounds are the most severe. Those are the ones that are where the flesh is exposed and they're red. We see lake trout have a much higher rate than Chinook salmon in Lake Michigan. Whereas in Lake Huron, the inverse is we see the inverse, where Chinook salmon have a much higher rate than lake trout. And this kind of information is important for the sea lamprey control managers and researchers to basically kind of sort out what the actual damages are from lake trout, or excuse me, from sea lamprey predation. So I'm going to cut it off here. I'm just going to have a few concluding statements. Coded wire tags have helped answer many questions to date and will continue to do so. And it's provided managers and stakeholders with information that we previously haven't had available and are used widely by the management agencies to make stocking decisions and other decisions. And the collaborations have broadened the program quite a bit and is value added for the resource. So thank you for your attention. I'm Chuck Bronte. There's my contact information. I'm also recognizing Matt Cornice, who helped put together this thing, but we have a slug of other people in our office that actually do the on the ground stuff, and they're too numerous to mention, but technicians, tagging trailer operators, they all make this possible, along with the angling public, creel clerks, and the hatchery staffs. Thanks for your attention. I'd be happy to answer any questions.
Well, thank you, Chuck. That was a very comprehensive presentation. And audience, do you have a question about trout or salmon in Lake Michigan for Chuck? So maybe just a definition thing, Chuck, when you were talking about steelhead and origins and stuff, you were talking about streams and stuff like that. Can you explain just a little bit, why would you expect there to be differences in ear bones from one stream to the next?
And that's all, yeah, the trace element profiles found in calcified structures, basically the ear bones of these fish, is basically controlled by the ambient chemical composition or the geology of the water that's, of the area where the animal is born. So those get incorporated early on in life. What we do, what the folks at Central Michigan do is look at the core of the eardrum and look at the chemical composition of that. That basically and then they analyze that over time and across different areas, and they develop a signature for a group of streams that share the same chemistry, hence, or the same geology, hence the same chemistry. And then we look for that same chemical composition in the fish when they're older out in the lake, which are now mixed throughout Lake Michigan, and basically are able to trace back where they likely were from, where they were born. So we're doing that for Chinook salmon as well.
Very interesting. Are there any other questions? Laura, are there any questions that are available through Facebook?
No, I have not seen any on Facebook, but I was wondering if the fish were not tagged last year, were they planted in the lake and will they appear to be naturally born? If they, if
so
they were there.
they were stocked out. We did begin tagging some of the fish in March, and then we had to pull back because of COVID. Very few fish were tagged. What we're gonna do for the Chinook salmon now this year, actually, let me backtrack. We did get into the federal hatcheries last year and coated wire tagged and adipose clipped all the federal lake trout. The Chinook and Steelhead were not tagged or marked, but we're going to use differential first year growth because the hatchery fish get, are larger when they go out than the wild fish at the same age. And we're going to look at differential growth rates of the first year to separate out hatchery from wild. That is, those fish with a faster first year growth, we will assume to be hatchery reared fish.
But it will be, and it does add I think so. But not tagging fish like you wanted to in this year will complicate things a little bit in the next several years, correct?
Without a doubt, yeah.
Right. And are you planning on tagging at full strength this spring?
Yeah, we are now tagging. We've already visited 3 state hatcheries so far. We're going to be marking all the Chinook with fin clips. We're going to be putting coated wire tags and fin clips in all the steelhead and doing lake trout later in the year. We are proceeding with the program as if COVID will cooperate. Then not only the tagging end of it, but also at the data recovery end of it.
So
we are currently onboarding technicians for this year, and we're at Wild Rose Hatchery right now in Wisconsin, and we're at Wolf Lake Hatchery in Michigan.
Very good. Are there any other questions? Okay, well, again, thank you very much, Chuck, and actually thank you to all you that have joined in on the Fishtail lecture series this year, on this particular one and the previous ones. We would really like to hear from you what you thought about the program this year. So if you could please drop us a line at fishtailslecture@gmail.com, that would be much appreciated. And thank you very much for joining us. And we will see you again next winter.